Hello, I'm Songyong from Seoul University, and it's a great honor for me to be awarded AThe Outstanding PhD Award. Now, I will briefly explain the results of my research. The chromatin-binding protein PHF6 functions as an E3 ubiquitin ligase of H2BK120 via H2BK12 acetylation recognition for activation of trophectodermal genes. PHF6 is a gene with two extended pil-length homodomains called EPHD domain for short. The PHD domain is a domain belonging to the zinc finger superfamily that maintains its structure by capturing two zinc ions, and the extended PHD domain has a structure that captures three zinc ions. The extended pH domains of PHF6 are highly conserved among species and has been thought to work in transcriptional regulation by recognizing histone modification. To date, the most well-known system for the role of PHF6 is in leukemia. Several mutations of PHF6 have been reported in T-cell acute leukemia patients, and many studies have shown that the functional loss of PHF6 induces TALL development. However, the precise molecular mechanism of PHF6 has not been determined yet. In addition, in several case studies, PHF6 mutations have been reported as an important factor causing Boysen-Forssman-Lehmann syndrome. The various symptoms of BFL syndrome patients are typical developmental problems, and this led me to assume that PHF6 will play an important role in the early development. Through these previous reports, I have conducted a study to find the importance and molecular mechanism of PHF6 in early stages of development. To see how PHF6 affects early development, I generated the PHF6 knockout cells from GHBTC4 embryonic stem cell line. I treated two kinds of chemicals to induce differentiation of embryonic stem cells into different lineages and compared the difference in the transcriptome between wild-type and PHF6 knockout through mRNA sequencing. As a result, I found that only doxycycline-dependently induced genes were not sufficiently expressed in PHF6 knockout cells. When checking the biological process in these genes, I found that placenta development, which is differentiated from trophectodermal lineage, belonged to the most significant biological process. Also, I confirmed that many trophectodermal marker genes were not sufficiently expressed in PHF6 knockout cells. To further confirm this, I compared and observed the differentiation of wild-type and PHF6 knockout cells by performing embryonic body formation. As a result, I confirmed that the cell layer extending out of the core of embryo bodies were not grown in PHF6 knockout cells. When checking the lineage of the outer cell by immunostaining with CDX2, which is a trophectodermal marker gene, I could confirm that this layer is a cell population that differentiates into trophectodermal lineage. Also, I was able to check that expression of trophectodermal genes were not sufficiently expressed in PHF6 knockout embryoid bodies. Taking these results together, I conclude that PHF6 has a transcription regulating function in differentiation of trophectodermal lineage. Next, I conducted an experiment to see how PHF6 causes transcription activation. I performed an in vitro peptide binding array to see which histone modifications are recognized by PHF6. As a result, I found that PHF6 recognized specific modifications of histone H3 and H2B. And by performing individual histone peptide binding assay, I conclude that PHF6 recognized histone H2B K12 acetylation. Next, I compared the sequences of the EPH domain 1 and domain 2 of PHF6 and found that excitic residues rich regions only existed in the EPH domain 2. When using a mutant without charge of this residue, I confirmed that the H2BK12 acetylation recognition function of PHF6 was significantly disappeared. Through this result, I conclude that PHF6 has the ability to recognize histone H2BK12 acetylation through the EPH2 domain. Next, I checked which histone modifications showed changes with or without pH6. As a result of comparing the levels of several histone modifications, I found that the level of H2BK120 ubiquitination were decreased in pH6 knockout ESL upon doxycycline treatment. Therefore, I checked whether pH6 has an ester ligase activity against H2BK120UB by in vitro ubiquitination assay. As a result, it is confirmed that pH6 has an acetylation-dependent ester ligase activity against H2B ubiquitination. I also found that ester ligase activity was disappeared even if only one of the ePH domains was destroyed. Through this, I checked whether ePH domain 1 of pH6 has an ester ligase activity and confirmed that only pH1 domain mutant failed to bind with UBCH3, which is an E2 enzyme. On the other hand, I confirmed that pH domain 1 mutant still binds to H2BK12 acetylation, but pH domain 2 mutant lose this binding affinity. Through these results, I conclude that pH6 recognizes H2BK12 acetyl through pH domain 2 and has an ester ligase activity through pH domain 1. Next, I checked whether this molecular function of pH6 affects H2B UV levels and gene expressions in the context of trophectodermal differentiation. As a result of checking through chromatin IP experiments, I confirmed that pH6 is located on the promoter of trophectodermal genes, and the levels of H2B UV and H3K4 trimethylation were increased in pH6 dependently. In addition, as a result of expressing pH6 wild type or mutants in pH6 knockout cells, I confirmed that H2B UV levels were not recovered in each mutant and that the expression of trophectodermal genes was not sufficiently increased. Taken together, I have shown that pH6 is a critical factor that regulates the transcription of key genes during trophectodermal differentiation. At this time, I confirmed that pH6 has both H2BK12 acetyl recognizing function and ester ligase activity over H2BK120UB through two extended pH domains. I defined the molecular function of pH6 and its functional role in trophectodermal differentiation, which has not been identified yet. The results of my research were published online in Nucleic Acid Research on July 31st and were officially published at September 18th. To close, I would like to express my gratitude to Professor Sung-hee Baek, my advisor, who gave me a lot of advice and guidance on my research during my PhD program. Also, I would like to thank the collaborators and lab colleagues who have contributed a lot to my research. Lastly, I would like to express my gratitude to the Research Foundation for appreciating my research and conferring great awards. Thank you for listening to my presentation.